DNA replication is the process in which DNA is replicated in preparation for cell division. Topoisomerase is an enzyme that untwists the backbone, and DNA helicase is an enzyme that unzips the DNA by breaking hydrogen bonds between the bases that forms the replication fork. Single-strand binding proteins bind near the replication fork and hold DNA apart so hydrogen bonds don't reform. DNA polymerase III then adds DNA nucleotides, which adenosine binds to thymine and cytosine binds to guanine.
So at the RNA primase, it's an enzyme that lays down the RNA primer, and the primer is the short section of RNA that polymerase can bind to. And so from there, it leads back again to DNA polymerase III, which adds the DNA nucleotides. And then from polymerase III, it goes to polymerase I, which removes that RNA primer and replaces it with the DNA nucleotides. Right next to polymerase I is the DNA ligase, which is the glue. It sort of acts as a glue that joins the Okazaki fragments together, leading towards the end of the strand.
The leading strand runs from 5' to 3', and it replicates from 3' to 5', and it's continuous. The leading strand requires DNA polymerase III. The lagging strand runs from 3' to 5' and also is replicated from 3' to 5', but it's not continuous. It requires RNA primase and RNA primer, and all this takes place in the nucleus.
DNA is a nucleic acid made up of DNA nucleotides. DNA is arranged in a double helix with a phosphate and sugar backbone held together by covalent bonds. The rungs of the double helix consist of 4 nitrogenous bases, guanine, cytosine, adenine, and thymine, which are held together by hydrogen bonds. Purines are the single-ringed nitrogenous bases guanine and adenine, while pyrimidines are the double-ringed bases cytosine and thymine.
Now you know all about DNA replication and structure. The end!